All right. In this video, we're going to look at DNA replication. DNA replication um, is uh, is going to happen during the interphase cycle. Well, let's look at it as we go through. Uh, DNA, deoxyribonucleic acid, which again is a nucleic acid. This is where our genetic code is going to be stored. And it is also the instructions needed for making proteins, right? Which again, as it points out here, includes the enzymes we've looked at. But again, any protein in the body, this is where the code to make it is going to be. Now, the gene is the portion of DNA that is the genetic code for making a single protein. And if I look at all of them together, we call that the genome. Now, the portion of the genome that encodes the proteins, it's only about 1% of it, is the exome. Now again, that means there's a whole bunch of it that is different, and we won't get into that, but this, as it says, the rest of the genome regulates the activity of genes for protein synthesis. Again, what it's saying is they control, there's the recipe, it decides which cells make which recipe, basically. A DNA molecule consists of building blocks called nucleotides. We looked at this in a previous chapter, all right? So DNA is nucleic acid, and the building block of all nucleic acids are nucleotides. DNA form a 2-sugar phosphate backbone, as it says, which talks about how it twists, and whatever this twisting ladder, we call it a double helix, as it looks in here, and you've probably seen it before. Now, the bases are really the key to this. This is for us to understand DNA and how we're going to look at it. DNA, the bases, which are the things that bind together, are these 4: adenine, guanine, cytosine, and thymine. And adenine and thymine will always bind together. We'll see this in just a minute when we look at another part of it. Guanine and cytosine always bind together. Now, they form their bonds by what we looked at earlier called hydrogen bonds. And I hope everybody remembers the most important element in a hydrogen bond is hydrogen. Yep. And so nucleotides of one of the DNA strand exhibits a complementary base pairing to the other on the other strand. Now, this is what I was talking about here. Adenine always binds with thymine, or vice versa, and cytosine with guanine. A to T, C to G. All right, now this is that double helix look, and you can see again anywhere there's an A on one side, a T is on the other, no matter which side you're looking at. And anywhere there's a G on one side, a C is on another. Doesn't matter which way you're looking at it. So this is how you would remember it. Now, I would probably, there's a part that I'll talk about on the test, and we'll look at it, where I will put some letters on one side. They won't have this drawn out, but they'll just be A, G, T, G, C, whatever. And then I'll ask you, tell me what's on the opposite side. And I'll give you 4 lists of letters, and you'll have to pick it out. Now, this video is about DNA replication. And what's going to happen is, during the interphase of normal cell life, and particularly the S phase, DNA is going to make a copy of itself. It is going to unwind, and each leg will have a new set of nucleotides build on the other one. This is done by DNA polymerase. It is what is going to be making them from one side to the other. Now, this is going to happen, and at the end of DNA replication, I'm going to have inside the nucleus 2 sets of DNA, so that when this cell decides to go through mitosis, each of the new daughter cells will have a complete strand of DNA inside of it. And this is a picture of showing how it's going to be splitting. It's far more complicated than that, but basically we're going to split. One parent leg is going to go this way, one parent leg is going to go that way, and we're going to rebuild these opposite counterparts on each side. And at the end, we're going to have 2 new, complete, and identical DNA strands. And that is DNA replication. I hope that makes sense. And again, have a great day.